There it is.
Oh, I just want to do anything.
Alright. Which ones do you want to go over, Mason?
Which ones do I want to go over?
Yeah. Do you have any specific ones that you want to go over?
Let's just check my answers. I want to make sure I got them all right. Did you get 1 out of 64 for the last one? What did you guys get?
Yeah, 1 over 32.
1 over 32?
Yeah, it's 1/2, 1/2, 1/4, 1/4.
Is it?
4, 16, 32, 64. It's 64. It's 1 over 64. I just did my math wrong. Good job. Sucker.
Where did I put my answer?
Holly, do you have anything to participate?
I don't. You have more of my papers.
Nice use of colors. What did you guys get for number 4? Mason, what did you get for?
For each of the following, identify the total number of chromosomes. 48. Duh.
Did I get the wrong answer?
Why do you have like 12 answers?
Because you have to state the number for all of those.
Hey, I'm back.
Oh, I see.
We're going over number 4.
Okay. Did we figure out number 7? It is 1/64th, right?
Yeah. Good job.
Okay, cool.
Math.
Awesome. Number 4. What'd you guys put for number 4?
96, 24, 48.
Is that what you got for the first one?
Wait, number 4?
It's easy now.
Yeah, number 4.
Something else. I put 48, 96, 48, 24, 48.
Wait, what?
Yeah, something different.
Wait, so you have 3 answers to each little bullet point, right?
Oh, shoot, I didn't even do that. I just did number of chromosomes and stopped reading. My bad. All right. So what'd you put for number 1, or for the prior to DNA replication?
I put 96.
I put 48, right?
The diploid chromosome number is 48 prior to chromosome.
I think that's what that means, right? Or I could be wrong, but
Holly, is it 48?
the haploid number is the number of pairs. I think I could be wrong. But
What are
you're
we?
100%.
What's
right.
the question for which one?
Okay, the number of total. Okay, check it out. Blah blah blah.
Well, I know, but
diploid number, and then the number of chromosomes prior to DNA replication. It's 96 though. No. I think
diploid chromosome number
48. Yeah.
is 48, so the first one's 48 prior to DNA replication.
Beginning of metaphase 96, right?
beginning of metaphase, yeah, 96, because the 48 have all replicated.
End of telophase 1.
48 again, because
End of telophase 2.
24.
After fertilization 48.
48 again, right?
Wait.
And then we got to put the number of homologous chromosome pairs. See, now we're looking for number of pairs, lovely, and number of sister chromatids present. Prior to DNA replication, there are 24 pairs, right?
Right.
And 0 sister chromatids. Yeah.
Prior to DNA replication?
Yeah, right? There are 0 sister chromatids, and there are 24 homologous chromosomes, right?
I thought if you had a homologous chromosome, you had to have a sister chromosome.
No, because homologous chromosomes are the set of chromosomes that line up, one from mom, one from dad. Sister chromatids are replicated
Yeah,
chromosomes.
so like this is a homologous. I can't even see that. This is a homologous chromosome, but this is a sister chromatid. So if you have 24, then you have to have 48, right?
Only if it's been replicated, right? Because there's no replication has occurred yet.
Oh, yeah, good point. So you own, but you would still have a sister chromatid.
No, you wouldn't have any sister chromatids. Right?
Then, no, I don't know. This is something I legitimately don't know.
I think you wouldn't have any because sister chromatids are the ones that are genetically identical after replication.
Okay. So you'd only have homologous pairs?
Yeah, so you'd have homologous pairs. That's the number of paired chromosomes you have, one from mom, and it's half of the diploid number.
The first one?
Number of homologous chromosomes prior, or number of homologous chromosome pairs prior to DNA replication. Yeah, I have 2.
24.
I have 24. Wait, do you have 24? Okay, and then the number of sister chromatids. So those
0.
only after DNA replication.
Correct. Okay. Because sister chromatids are the copy.
Okay, so it's 48, 24, 0.
Yeah, that's what I think.
Okay, so nice. So awesome. Yeah. Beginning mark of 96. No, we both on 96.
So for part 2,
Beginning of metaphase 1. It's
48.
definitely 96.
It's definitely 48.
Definitely 96.
It's definitely 48.
definitely 96.
Definitely 48.
Definitely 96. You have to
Number
multiply.
of chromosomes?
The sister chromatids are multiplied. That's what's 96.
I think they're also
number of
96.
chromosomes.
Yeah. It's 96 something, then another 96. I don't know what's in the middle. Because I don't know how it's counting.
96, 48, 96.
Definitely 48, 24, 96. This is straight from a genetics teacher. I'm not gonna, like,
Okay.
this is correct.
Wait, so did you have someone help you with it?
I've already taken the class.
Okay.
No, I try to figure it out at 3 o'clock in the
I
morning.
can't see how the number of chromosomes would be less than the number of sister chromatids because sister chromatids are chromosomes. I could be wrong, but I don't understand it if I am.
Because the number of chromosomes has to double so that it can be replicated and each chromosome has 48. Because if that were true, then each chromosome would only have 24, and that doesn't happen. It doesn't happen.
I'm pretty sold
Don't
on
take it up with the Brother DeWall. He's the one that gave
DeWall.
He's like the best geneticist.
He's the best at everything. I think it's 76. Number of homologous chromosomes.
The only thing I'm sketchy about is the number of homologous chromosomes because I don't know how that's counted.
I put 48, but I think it's 24, right?
24. I mean, it just depends on how it's counted. Basically, do the replicated ones count as new pairs? So now you have 2 of the same pair. Do you count that pair twice or do you only count it once?
I counted it twice for the first time, and then after talking with you guys, I feel like
I don't know. I don't know how that's counted. I have absolutely no idea.
let's see how it's. Do they have a key to this one or no?
I wish they did.
Okay, so tell me how it's 48.
Or beginning of phase 1.
Yeah.
Because the number of chromosomes there's no change. There's no separation. There's no change prior to metaphase. You have a DNA replication.
But prophase comes before that, and DNA replication happens.
Yeah, DNA replication's already occurred during metaphase.
Yeah.
I don't know how to explain it.
Okay, next question.
Okay. What are we going with? I mean,
I'm keeping 96.
I'm going 96, 48, 96, but I need to talk to the teacher or something because
That's
I don't.
exactly what this is going for.
I'm going to ask her if she has a key or something.
24, 96. Or 48, 24, 96.
I am sticking with this.
Okay, now what about this one? This one I'm actually kind of
That was
confused about.
awesome.
This wasn't an assignment. This was lecture
Oh,
from the teacher.
and he like wrote that up on the board.
Yes.
No, no.
What's up?
Okay. What's the next one?
End of telophase 1.
Number of chromosomes is 24. Number of homologous chromosome pairs
Wait, number of chromosomes.
is not existent.
Wait, which one is non-existent? Okay,
Homologous.
so
There are no more homologous
Yeah, there's
chromosomes.
no more homologous. That's 0.
that makes sense to me. But there are sister chromatids.
There are still
Yeah.
sister chromatids and there are still chromosomes.
Wait, that makes sense to me. Okay, wait, but I have how many number of chromosomes is 48.
Yeah, I have 48, 0, 48.
Yeah, because this sounds like he's going off of metaphase 2, telophase 2.
It's not, because this is telophase 1, this is telophase 2.
I know, but if this were true, then if at the end of telophase 1 you had 24, then that means you would only have 12 in your gametes.
No, this is individual cells, so you have 24 in each cell, because you have 48 in one, and then they split in half. And so there's 20.
No, there's 48, and then it doubles to 96, and then it goes back down to 48, and then
Goes
it
down to 20.
replicates there to 24.
Yeah.
I'm
I
not
agree
going
with
to argue with you.
No, that's really no. Those are the steps of meiosis.
Yes, I understand what the steps of meiosis are, but this is what I have.
The number of chromosomes can't be smaller than the number of sister chromatids.
I don't have anything to say. This is what I have, and this is what I'm sticking with. You're not going to change my mind.
I know.
It's fine.
We're not trying to change your mind. We're just trying to understand your problem. That's all. Or like what you have, because you were in a lecture, and so we're like, wait, how is that?
I don't know how to explain it. I'm not good with these kind of things, but
But for sure, those are the answers.
this is legit what the teacher wrote on the board.
Mason, what do you think?
What'd you say?
Either I'm super missing something, or DeWalt was wrong.
He's not wrong. I doubt the teacher would be wrong.
I'm
Why?
very up to the possibility that I'm just confused, but if I am confused, I need to understand why.
Yeah, exactly.
I understand.
I'm going to send an email to the teacher just asking her for like a key for that question and telling her we didn't quite understand it. And because we're not even turning that question in, so.
No. Okay, so in this one though, the sister chromatids would be 24. Right? At the end of telophase 1, the number of sister chromatids would be 24.
Which one are we doing?
End of telophase 1, and the number of sister chromatids.
Number of sister chromatids should be
48.
right? I put 48.
So sister chromatids is synonymous with chromosomes.
Yeah. For now.
The,
What did you say?
this one. Oh, sorry. This one.
For now. Okay. But once we go through telophase 2, that's not true anymore.
Sister chromatids. And then I asked, is sister chromatid synonymous with the word chromosome. But after we go through what?
Huh? After we go through telophase 2, it should be 2400.
That's what Allie has too.
And then after fertilization, it's back to the beginning, 48240. I'm just going by the definition off of your key question. I mean, because the only way 16 works is if you're counting sister chromatids as individual chromosomes.
Okay, here. Telophase 1 in cytokinesis. The members of a pair of homologous chromosomes have completely separated from each other, and cytokinesis produces 2 cells. Each of these 2 cells contains only one set of chromosomes, even though each chromosome is composed of 2 sister chromatids. The 2 cells are considered to be haploid at this point. Thus, meiosis 1 converts a diploid cell, 2 chromosome sets, into haploid cells. Each cell has 1 chromosome set. Finally, the chromosomes may decondense and the nuclear membrane may reform at the stage.
Yeah.
Can we go over some general vocabulary? This may seem redundant and frivolous, but I still want to go.
Let's do it then.
Okay, so
So that's why at telophase 1 you have half the number of chromosomes, but still the same amount of sister chromatids.
I disagree.
I don't understand. There's not one person who doesn't understand it.
It's per cell, right?
Right, but you're going from
96 only works if you count each pair, and half of those pairs are now in a different cell.
I don't know how to explain this.
I just want to go into Dual and see what's going on. Okay, so chromosome set is the
Because-
maternal. Oh.
This is a chromosome set, okay? When
Yeah,
you
it's
have
the maternal and the paternal, right?
Yeah.
Wait, I thought this was a chromosome set. And these are sister chromatids.
this is a chromosome.
Mm-hmm. I understand
These
that.
are sister chromatids.
I understand that.
Okay, so if you have this 2, 1 in each.
These are homologous pairs, and this is a chromosome set because you have a maternal and a paternal.
Right.
Yes.
These cells each contain only one set of chromosomes, even though each chromosome is composed of 2 sister chromatids. The 2 cells are considered haploid at this point. Thus, meiosis 1 converts a diploid cell, 2 chromosome sets, into 2
haploid cells?
into 2 haploid cells, each that has 1 chromosome set.
Yeah.
The thing is a haploid times 2. That's what you're saying. That's what that says.
What meiosis is creating is you're taking a diploid cell, so it has one from, you have your sister chromatid, your chromosome, your sister chromatids, and your chromosome, and you're separating them, creating cells that only have one of each, not both of them in the same cell anymore. They've separated into 2 different cells.
Yeah.
So you cannot have double the amount of chromosomes in each of those cells
So
still, because it's splitting it in half. It's literally taking the cells and breaking them in half.
So what does prophase 1 do?
Prophase 1 is condensing everything
And replication.
in
Cross-
replication.
So where does that replication come in then?
Replication's before then.
Did. That's
Yeah,
what
I
it's
know.
creating. I
Right.
can't drive. Prophase 1. In addition to the events detailed above, the nuclear envelope begins to break down during prophase 1 and cytoskeleton structures called the meiotic spindle begins to form. Prometaphase 1. During this stage, the meiotic spindle has completely formed. The paired homologous chromosomes attach to opposite spindle poles via the microtubules in the meiotic spindle. The nuclear membrane is completely disassembled stage, metaphase 1. At this point, the homologous chromosome pairs are aligned along the metaphase plate, imaginary plane halfway between blah, blah, blah. Homologous chromosomes are aligned as pairs, forming a double row in contrast to the single row of chromosomes observed in metaphase of mitosis and meiosis 2. So, I'm not good at explaining this. I'm sorry. I wish I would, but I'm not.
Okay, so what I'm hearing, like just for clarification, is that you have a cell and then it splits and you have like you have a haploid times 2 and then you just have a haploid. And they're not calling it a diploid, they're calling it a haploid.
Yeah.
I've been taught that too. In a weird
So
way.
if you can see what the difference is here, it's really teeny. So you have, you have homologous pairs still, and it's separating them. And then you go even further, and you separate those.
homologous pairs, your sister chromatids.
The sister chromosomes are separating.
Where's your thing again? Your chart?
There are no.
40.
I don't know how to explain that. I'm sorry.
I just don't get how you have the same number of chromosomes after telophase 1 and telophase 2. Did you send that email to that teacher? Mason,
What'd you say?
did you send that email to the teacher?
Yeah, I just finished typing it. I'm sending it now. I basically sent her the problem, told her we were confused about it, asked her if there was a key, told her kind of which part of it was confusing us, and that's it. I sent her the problem, so. Also, no sweat, but it's Macon, hard C.
What did you say?
I said my name's Macon, hard C.
Your name's Macon?
Yeah.
Sorry. I've been calling.
No worries.
Do a lot of people call you Mason?
I've heard that for sure. It didn't like bug me at first because I mean you said Mason, I was like, oh whatever, like we're talking about bio, it's not important, you know. But
I've never heard that name ever.
I have.
Oh, yeah, yeah.
Yeah, I'm not too worried about it, but. So,
Oh, I was like, what are you watching right now?
do we have any,
I pulled this out.
Okay, let's see here. Number 5, did you get 1/2 as tall and 1/2 are dwarfs?
yeah, and same for the genotype. 1/2 are heterozygous, and the other half are homozygous.
Yes.
Oh wait, sorry.
And then,
Right.
let's see here.
You've got 0 that are that are homozygous tall, and then half of them are heterozygous tall, and half are homozygous short, right?
On 5, yeah.
Yeah. Okay, and then 6. I got 3 to 3 to 1 to 1.
3 to 3 to 1 to 1. I didn't do the cross yet. I just did a homozygous recessive with a heterozygous crossed with 2 heterozygous. Yeah.
Wait, what?
What did you get? I'll just do it real quick.
Because what I got was there's going to be some offspring with every phenotype, and that was the ratio. But
Did you have this one last semester? No memento. Macon, where's your name from?
family history,
That's pretty sweet.
Mormon parents. I'll do it to you.
Mormon parents for the win.
Yeah, there's actually a city in Georgia named Macon. It's
Really?
pronounced the same way I am. Yeah, it was actually named after the same dude that I am.
How did you know that?
Because I know people from Georgia.
I've never been there, but
Evidently, that's pretty sweet.
So, what other predicted phenotypes?
all right. So,
Did you do that one? I'm just thinking.
what did you put for 6?
Give me 2 seconds.
Either of you.
Memento. Sorry.
Okay, what did you get? What are the protected phenotypes?
Do you guys have an answer for this one, or?
No, yeah. I have some are gonna be yellow.
Are you doing it?
Well, it's gonna be every phenotype possible. So yellow smooth, yellow green, constricted green. Do you have that?
Yep. I got every phenotype in a 3 to 3 to 1 to 1 ratio, but
Wait. Does it ask for the ratio?
no.
Nope. Going above and beyond. And then 1 out of 64. Sweet. But you could do it in 5 seconds.
What?
The problem.
Oh, I don't care to do it.
Yeah, okay. Alright. Do you want to go over this? Can we go over this?
No, you go over it.
Megan, do you care if we go over the applying genetics problems?
No, go for it. I'll work on it in the background, and if you guys, I mean, I guess I can ask questions. Are we turning in the applying genetics thing tonight?
Yeah.
Are we just uploading a Word document or something, or?
It's like just 3A, right?
So what it is you do the problems and then you just there's the problems and you're supposed to do them, and then you're supposed to attach like a picture or whatever, a scan of what your work or however you're gonna do it, like a
I'll just
document.
do the Word document
You
that's on, and then
Oh, question number 3, part A. That's the only question you have to put in.
of the applying genetics problems.
Correct. And then there's a key review, which is like, just like, did you do the problems, basically, I think, right? That's what it is.
Okay.
And then there's a quiz.
So
Yeah.
we're supposed to do the whole thing, but we only turn in question 3.
Correct.
Yeah. Yeah, you're right and all that. And then the key is the key to this.
This is. But there's like a start.
Oh.
It's a you get credit from completing
Oh,
the
yeah,
survey.
yeah. It's like just asking you if you did all the problems.
Yeah. That's what it was the last time. What else do you have questions on this?
What is that question on?
What else? Let's skip that one first. 2E.
Oh, wait. Okay, just kidding. No, questions with that. She just keyed it with null. And hypomorphic, but I put it recessive and dominant. But
Oh.
those are just synonymous for each other.
What?
recessive and null. Allie, you're supposed to be the one that's helping me get an A in this class.
In the class right now?
Do I have an A in this class? Yeah, we'll have an F in 100%.
That's all you have questions on?
Yeah.
But did you do 3, 4, and 5?
I think I did 4. 5 questions, 1, C, D, and E.
On number 4?
And
Horse?
I didn't do 3 and 5.
Okay. A horse has 32 pairs of chromosomes. Okay.
Yeah.
How many sister chromatids are present in a horse cell during prophase 1 of meiosis?
64, right? No.
It's going to be double.
Pretty sure. It's 128.
Okay. How many chromosomes are present during prophase 1 of meiosis? It's going to be exactly the same. So if you have 32, you're going to have 32.
But the answer is 128.
Just kidding. I lied.
You have 32 pairs of chromosomes, so you really have 64 chromosomes, so then it's going to double, and it's going to be, 128. Right? Yes. Bacon.
Hey, what's up?
Okay, 4C.
Are you guys doing that one?
Yeah. It's 128.
Okay.
Right? And the reason that is
It's
because,
what?
okay, so you have 32 pairs of chromosomes, which means you actually have 64 chromosomes.
Yep.
You would have 128 sister chromatids after prophase 1 of meiosis, and it's asking, yeah,
You're saying C?
yeah.
Yeah, I agree with that.
Okay, sweet, that's the answer. How many, okay, then how many sister chromatids are present for a horse cell during metaphase 2?
Wait, hold on. During metaphase 2 or at the end? Okay, so during metaphase 2. Okay.
So
I guess
it's
it won't matter if it's the beginning or end. So
32. The answer, 64.
yeah, it's just half. Because metaphase 2, it hasn't divided the second time yet.
So, well, I'm almost positive.
What?
Oh. Okay, and then how many bivalents are
Five
present for horse? Okay, what is that one again? What is bivalents?
That's a pair of homologous chromosomes.
So 0. See?
During anaphase 2, yeah, I think 0. Right?
Yes.
Anaphase 2, they've all been split up. Everything's been split
Okay,
up.
so at the end of that, you don't have any homologous chromosomes or sister chromatids, right?
Yeah, at the end of anaphase 2, everything's already split up.
And you get rid of your homologous chromosomes at the end of telophase 1.
Yeah.
Yes. Okay. Go team, go. I don't have any other questions. Okay. Do you want to go over anything else? Megan, do you want to go over anything else?
I don't think so. Do you guys want me to send you guys an email when she responds?
Yeah, just CCS on it.
Okay. I don't think I have your email, Allie, unless maybe it was part of the original. Oh yeah, I think I do actually. I just never saw any responses from you.
Because I never saw you.
Cool. Yeah, I've got it. So I'll just send an email to both of you. But yeah, that one dude never showed up. So is there a place where we kind of do we just put whoever participated or?
Well, what we're gonna do is we're just gonna link this video to,
To the conversation group board.
there's a discussion board that we're supposed to link it to.
Yeah, but there's no way she's gonna watch it unless there's some sort of a dispute.
Right, and so what we'll just do is we'll say these are the people that attended.
Yeah, okay, sure. Sounds good. Who's linking it? Are you, Tylen? This is your computer, right?
Yeah, I'll just do it.
Okay, cool.
Alright, well, I'll see you guys next week.
Yeah, send out an email or something. Do you guys want to try to do it earlier next week?
That's fine.
Yeah.
When are you guys free?
I'm free afternoon and evenings.
I'm not free
Yeah,
meetings.
I'm done with all my classes by 1 for the second half of the week.
I
I'm
can
done
do
by
that.
2.
Yeah, you can do too.
All right, cool. So just send in an email, but I'm down to hang out or do the problem sometime between 1 and late. All right, cool. Thank you.
Yep,
We'll see you guys.
Have a nice one. Bye.
So push stop.